which are going to be blotting techniques. So this, you can see Western blotting. We're going to go into that. And the first one I wanted to talk about was Northern blotting. And so Northern blotting is used to detect specific RNA sequences. And it can also be used to measure the RNA expression for particular genes. And so this is a diagram on the right as to how a Northern blot, like the, I guess, workflow of it would be. So we have the sample here, and here is the RNA extraction from the sample. Usually the RNA will still be in, will still be like a double-stranded RNA in a lot of these samples, and so that's why when they are separated, so that's why we have the denaturing agent of formaldehyde to break up those interactions, to make sure that when we are separating the RNA by gel electrophoresis, we are only getting those single-stranded RNAs in the event that they, hybridize with each other. And so then the second step is that the RNA strands from this gel are transferred to another surface, which is usually of nitrocellulose or nylon. And that surface is known as the blotting membrane. And so this process of transferring the results from the gel to this other surface is why all of these are called different types of blots. And then after it's transferred to that blotting membrane, the RNA molecules are then exposed to probes that have a complementary sequence to bind with the specific RNA sequences that we are looking for. And then finally, the blotting membrane is then washed off so that any excess probes are not going to be on that blotting membrane anymore. And usually these probes also have a reporter that show a color or radioactive signal. And so that is going to be looked under, like they're going to be visualized under either X-ray or some other types of visualization techniques associated with that reporter of the probe. And then that's where we would be able to see, the specific, how much of the specific RNA sequences that we have. So on the flip side is the Southern blot, which is pretty similar to the Northern blot, except that instead of detecting specific RNA sequences, the Southern blot is detecting specific DNA sequences. And additionally, we have restriction enzymes that are used in the Southern blot to cut DNA fragments into smaller pieces. And so restriction enzymes are used to specifically cut based on the restriction enzyme palindromic sequence. And so they cut at specific sites to break up these DNA fragments and to create these DNA fragments. And the steps in the process, as I said, they're pretty similar. So the DNA fragments are separated by size by gel electrophoresis. And then the second step is that DNA strands from the gel are transferred again to another surface, the blotting membrane surface. And then the third is that the DNA molecules are exposed to probes with a complementary sequence with the specific DNA sequences that we're looking for. And then the blotting membrane is washed, and then the probes have a reporter to show a color radioactive signal when visualized, and then that's looked under based on the type of visualization technique that they use. The Northern and Southern are pretty similar, just RNA versus DNA. And then we have the Western blot, which is a lot more, there are a lot more steps instead. And it's a laboratory method instead used to detect specific proteins. So the steps in this process, the first one, they're separated by size by gel electrophoresis, and then these protein strands are then transferred to another surface. So the blotting membrane surface as seen in the northern and southern blot. But then the difference starts from step 3, where the membrane is then blocked by buffers. And so common buffers are milk, normal serum, some highly purified proteins to make sure that the different, charges of the proteins and all the different properties of the proteins aren't quite interfering with the next steps that we want to do. So then the next step is that we wash to remove any excess blocking buffers. So common washing solvents are the tris buffered saline, TBS, and phosphate buffered saline, PBS. And that's just for any of the blocking buffers that are not blocking any particular proteins. And then the proteins are exposed to first primary antibodies that bind to the specific protein that we are looking for. And then the membrane is then again washed to remove any excess primary antibodies. And then we also have a second set of secondary antibodies that are added that bind to the primary antibodies that were originally there. And then we do another wash step to remove any, so this is actually supposed to be any excess secondary antibodies. And then after that, the membrane is visualized from secondary antibodies that have that extra light producing substance or radioactive substance. And then we visualize the membrane based on the type of visualization technique that we use.